Okay, thank you so much. Thank you for inviting me. Thank you for hosting me here. My name is Tapomoy. I go by Tapa. I'm at NCBS, Bangalore. So today we're going to talk about how confinement affects cellular growth in 3D viscoelastic environment. So before I start, I want to give shout out to Sri, who is a fourth year graduate student in my lab, who did mostly the whole heavy lifting of this project, along with Minakshi, who used to be a master's student. We collaborated with Professor Deepa Agashia's lab, who is a colleague at NCBS, and Pratibha is a postdoc in Deepa's lab, and we worked with Christina, who is a long-term friend, and she is leading her group in MPI-PKS now. And we had an opportunity to talk to a lot of colleagues in NCBS and outside. This work have recently came out in NatureCom. So if you're interested to read more about it, please feel free to. Okay, now that's outside the scope. Let's talk about bacteria, right? So Leeuwenhoek first looked at bacteria in 1677, if I'm not wrong. And since then, we are studying bacteria. But we need to kind of like focus on where Leeuwenhoek found this bacteria. Where did he see these bacteria to start with? He got this bacteria from different parts of water, but he also get it from blood, semen, saliva, sometimes some substances from tooth, any decomposing matter, all sorts of complex material where he sourced this bacteria from. But since then, our major understanding of bacteria and its biology itself, or how they interact with each other, all of that has been in these two kind of systems where these are homogeneous environment. Either you have a soft substrate of agar where you streak out this bacteria and you look at their colonies, or you homogeneously mix into them some sort of a broth and then you study how they are growing, how they are behaving, all of that. The question that always fascinated us is how do this microenvironment and their properties affect bacterial growth or their survival? But this question is not new. This has been there for several years and decades, actually. And this has been studied mainly from the perspective of a chemical composition of the microenvironment. Either you looked into uh, whether different kinds of carbon sources, how they affect the growth patterns of the bacteria, or we have looked into how different chemical environmental stresses in terms of antibiotics are affecting the bacterial behavior, their growth, their survival, their persisters, how they come and go, all of those kind of questions. We also know the interaction between different bacterial communities through small molecules, like how they exchange nutrients, or they create toxins which causes predator-prey kind of interactions, or some sort of mutualism in this kind of complex communities. Uh, but these are all small chemical molecules. In fact, when we looked into different colony architecture on 2D surfaces, these work actually used different agar concentration. They varied the agar concentration along with the nutrient concentration, and they found that the colony architecture are different. But if you look closely, most of the drastic difference of colony architecture comes towards the nutrient axis. So when you vary nutrient there, you can vary the bacterial colony architecture drastically. So now, having said that, we wanted to understand where do bacteria live. They live in this complex granular porous environment. They live in soil, lung tissues, or any sort of tissues actually, in viscoelastic mucus. This is their natural niches. This is where they live, right? So now my question is how the change in behavior of this material properties will change their growth or their fitness or their dynamics, right? Think about soil. With or without rain, the soil has different material property. So is your gut mucus. Depending on how much coffee you drink this morning, you will have like different the stiffness of your mucus. So what we wanted to know that how do the physical properties of these 3D microenvironment affect the colony growth of bacteria, right? So what we wanted to do then create something that mimics some natural 3D microenvironment of bacteria. Here we are focusing on mucus. Why mucus, right? Mucus is somewhere bacteria are naturally growing, and we would be able to isolate this bacteria from this 3D natural environment without risking ourselves that we are working with something that may or may not have evolved in laboratory settings, right? So if you look into the mucus, first we would like to create the structural property of mucus. Mucus has two sets of different pore sizes. Either it has a nanometer scale, few hundred nanometer scale, small meshes, which is mostly the pores, but you also have really micron scale large pores, which you can also see in the SEM images. So what we do, we take some small hydrogel granules which has an internal mesh size of 100 nanometers, and we swell them directly in liquid LB, which is a nutrient broth, and we pack these granules together very closely so they create these kind of micron scale pores which are order of few microns, and here bacteria can move or they can live and they can navigate through, right? So it has these 2 degrees of pore sizes, and because these are extremely transparent, as you can see through the liquid and the gel almost identically, you can use a lot of optically transparent nature of this gel to do a lot of standard assays like optical density measurements and stuff. Now, since we match the structural property of the mucus, we can also match the viscoelastic property of mucus. There are several studies which have collected the different viscoelastic property of mucus, and we found that we can match these viscoelastic properties by changing a small fraction of this hydrogel granules that we disperse in this liquid LB broth. Right. So by doing that, we can create 2 different degrees of confined 3D matrices, which matches the upper and lower limit of this viscoelastic properties that we found in the previous studies. And also, this gives us 2 different degrees of confinement. So one is a high stiffness, which has a less amount of pores or smaller pores. We will call this a high confinement matrix. And another one has a low stiffness, which has larger pores, which we'll call them like a low confinement matrix, right? Now, if I put bacteria in this system, we have to use something which is not evolved on a laboratory scale. So what we did, we isolated bacteria, at least 7 different types of bacteria, and these are all across the scale. They are facultative aerobe, they are gram negative, they are gram positive, some are motile, some are non-motile, some are long, some are short. So complete mixed bag of bacteria from the mucus of the gut mucus of the red flower beetle. This we did with our colleague Deepa Agashe and their lab. So once we get this bacteria, we exploit the transparent nature of this gel, which allows us to directly mix this bacteria inside this 3D growth matrices, and we can characterize their growth dynamics by looking into the optical density at some 600 nanometer, right? So now when we do that, and we start to see that as we increase the stiffness, as we change the stiffness of this gel, we start to create this like really, really distinct growth patterns. In many cases, we see as we increase the stiffness, we decrease the total amount of growth of bacteria in these gels. And in some cases, it's more, some cases, it is not that much. So what is going on here? Right? So one idea that you can think of, that high confinement reduces the overall size of the cell. Somehow the confinement is changing the cell shape. And since I am doing an optical density measurement, my shape change will reduce the overall absorbance, right? Or you can also claim that higher confinement can increase higher osmotic pressure. And when you do that, this elevated osmotic stress is drastically changing the cell growth, lowering the overall optical density. Or you can simply say like, look, your OD is a balance between overall growth and death. Maybe you have higher rates of death in your stiffer matrices, and that's why your OD is going down. In order to understand that, what we do is like we measured the different parameters of cell morphology, area or aspect ratio or their length, and we found that there is not much drastic difference between the cell shape in between these two different confinements. We did some viability analysis by staining for the dead cells into the stiffer matrices, and we found that in the stiff matrix, cells do not die as much as we thought it might actually happen. The cells are more or less very well viable in this system. And finally, we wanted to know if higher osmotic stress can cause this thing. So we created this, we supplemented our liquid growth culture with some PEG, which is a short chain polymer, which is increasing the overall osmotic stress by a factor of 1,000, and we found that even beyond what they are experiencing in our gels, and we found that there is not much of a significant effect of osmotic pressure in this range, which can cause drastic change in the growth pattern. So these are not the reason. So what is going on here, right? So in order to explain this, we thought of defining a parameter, which is the relative biomass ratio. It is nothing but measuring the overall optical density in any time point in the stiffer matrix, and we divide this by the softer matrix OD at that time point. And we find that if we plot all of them, they start to plateau after a certain time, and we take the end point, which has already plateaued. When we plot them, we start to see two distinct groups of bacteria that start to pop up. And these are, some of them are efficient performers, some of them are poor performers. What do I mean? If this relative biomass ratio, change in the total biomass, is less, which means that as I increase the stiffness, my overall bacterial biomass actually goes down much more drastically compared to a softer matrix. These are poor performers, and in the efficient performers, this ratio is high, which means they are not much affected by the increase in stiffness. So now, what is going on? So if we go back to our matrix over here and we look into our bacterial population and how special they are, we start to see that some of them these bacteria, which are poor performers, actually all of them are non-motile, and some of them are motile, which are efficient performers, right? So that makes sense in some ways, right? If your efficiency is very high, then that means that you can move very fast, and you can also explore a lot of different places, and that helps you to actually grow better. However, in order to understand that, what we did, we went back to a laboratory strain, which we thought at the first point that they might be sort of like evolved in the system. But now if we want to test the effect of motility only, we went back to the laboratory strain of E. coli. We take the E. coli and then grow them in this confined matrices. And then we also take a similar E. coli where we have deleted the flagella by deleting this flagellar gene. So it doesn't assemble the flagella, it becomes non-motile. So when we check them and their relative growth ratios, we start to see that they are both very efficient in growth, and the motility does not actually affect that much in their overall growth pattern, right? So it is not the motility. So what is going on here? So if we go back to the same disk where we have all the properties of the bacteria, we start to see that the poor performers are actually low aspect ratio cells. They are almost spherical, and the efficient performers are almost like rod-shaped bacteria. So why that is important, right? So shape does matter, and shape can matter in many different ways. One example is, if you are rod shaped, you are less contact inhibited, means like you can push your progenies further and you can separate out from your progenies or the neighboring cells much better than like a spherical colony. But also there is another promise in having like a elongated cell, is that elongated cells can produce a elongated colony, which will be less nutrient limited inside their core. Whereas the spherical cells will produce a spherical colony, which will be much more nutrient limited inside their core. In order to understand that, what we do is, I'm going to skip this one, which basically says that you have more actively growing bacteria in a shell of 10 microns, which is the nutrient penetration zone in a colony of different shapes. But if you actually think about that, so what we wanted to do is we wanted to simulate this process, keeping all biological processes same. So what we do, we take a high aspect ratio cell, which grows in their parental direction. So when they divide, all the both the daughter cell will have the same direction as their parent cell. However, for the spheres, we know from experimental results published previously that when the cells divide, they either divide at a perpendicular direction from the previous division, or they divide in the random direction. So in our simulations, we put them as a randomly dividing in random direction kind of approach. So when we do this, when we simulate this, we see indeed that as expected, these elongated cells will produce elongated colonies, and their colony morphology will be drastically different than the spherical ones, and which we can capture by their shape factor. So it is true that elongated cells in this kind of confined environment will produce elongated colonies. Now, does this actually hold under the experimental conditions? If we let E. coli grow under this extreme confinement, we start to see that indeed they create this extremely complex branched out elongated colonies, right? And if you take another naturally spherically occurring cell and you grow them inside this kind of matrices, you will see they will make more spherical colony. Now, I motivated the simulations by saying that you can keep all the processes, biological processes, same. Means your growth rates are same, your interactions are same, all of those things are same, right? But here I'm showing you two different bacterial systems. One is E. coli, one is E. faecalis. These have completely different biology. So how am I going to compare these two? So if I really want to do that, I need to make a raw-shaped E. coli into a spherical E. coli. So we can actually do that. We use a drug called A22. A22 is important because it messes with the MREB. MREB is a polymeric actin-like polymer in bacteria, which sits on the cell membrane and which is responsible for determining the shape of the cell. If you depolymerize the MREB, it actually becomes a sphere, and this sub MIC concentration of this drug A22 exactly does that. It does not affect the growth rate by a large, but what it does, it actually allows us to grow this E. coli into a spherical form. So now, if I can create the spherical E. coli, I would expect it to grow in a spherical colony, like I saw for E. faecalis. That's exactly what happens. If you take the spherical E. coli, you grow them under confinement, you start to see they form these beautiful spherical colonies. And if you measure their shape factor also, you will start to see that rod-shaped bacteria form really elongated colonies, whereas the spheres make really spherical colonies, right? We have measured many colonies, and that's what we find, that your bacterial shape dictates the overall colony shape. Now, why does that matter? Now, if you measure this relative growth of this E. coli in presence of, which is a rod-shaped bacteria, in presence of A22, which makes a sphere, you will start to see this as you increase the confinement, their performance drastically drop. They actually start to club with the poor performers, which happen to be the spherical cells. So I took a rod-shaped bacteria, I mess up its shape regulation, but it does not change the growth rate. But when I put them in a confinement, the increase in confinement drastically changed their overall growth performance, right? So the shape is actually a primary determinant in this whole process. And matter of fact, if you take a cell, which is take a bacterial cell, which is basically a sphere, under starved condition, but when they start to grow, they grow like a rod. These are Paracoccus sanguines. So if you take these cells and you grow them under different confinements, you will measure their relative performance. They start to club with the rod-shaped bacteria. So it doesn't matter what your inherent shape is. If you start to grow as a large aspect ratio cell, you will enjoy some sort of benefit of some sort of selective advantage over the cells which are growing as spheres. Now, I told you about selective advantage, how it affects, how it benefits, all of that. But if I want to claim them, I need to show how this advantage is giving these rod-shaped cells some benefits, right, in a co-culture system. So what we do, we set up a competitive co-culture where we have 50% of both shaped cells, and these are across different biology of the system. It is sort of agnostic of the whole biology of this bacteria at this point. You can take any rod-shaped cell, any spherical cell, mix them up half and half into this kind of two different stiffnesses, and you measure as I increase the stiffness, which population actually going to dominate compared to the softer environment. And what you find that in every single cases that we have studied, when you put a rod versus a sphere into a competitive co-culture, agnostic of the biology of their system, what turns out that as I increase the stiffness, the rod-shaped cells always win over the spherical cell. However, if you take two different rods and put them against each other, you will start to see there is not much of a competitive advantage any of these cells enjoy over another. Now, you may ask, like, what about chemical composition? What about one winning because there is a beneficial nutrient there? Our system actually does not stop that. You can have all sorts of chemical crosstalk. You can have like predator-prey interaction. You can have like mutualism in this system. However, all it says that when you increase the confinement and the physical properties of the confinement will have a say in which shape bacteria will dominate more than the other. So in short, what we know that if we measure all the aspect ratio of bacteria and we put them in a single phase space where we measure the single bacterium aspect ratio and relative biomass, we start to see all the rod-shaped bacteria club into one corner, all the spherical bacteria club into one corner, and there is a clear demarcation between how they occupy these two different zones of this phase space. So in long story short, what is happening here is bacterial shape sets colony morphology, and morphology dictates the future growth dynamics. And these future growth dynamics are favored towards the elongated colonies, which is formed by the elongated cells. So the rod-shaped bacteria enjoy a little bit of more beneficial advantage towards growth. Uh, it is very interesting that it does not invoke any sort of epigenetic changes. It does not invoke any sort of specific biology of the microorganisms. It does not also invoke any sort of any sort of transcriptome level changes either. So all we see is that pure interaction with the physical environment can also lead to some sort of selective advantage in bacterial growth. And finally, before I end this talk, I would like to leave you with another recent work we have done where we have also found that the physical confinement can dictate a transition in nematode motility. And it is also agnostic of the touch sensory neurons, how worms actually understand their environment. So this work is currently in bioRxiv. If you are interested, please have a look at that. With that, I would like to say thank you for having me here, and thank you for listening to me. Thank you.
All right, let's thanks Tapa for the wonderful talk. So I can see in the chat we have a question from Ashok. Is there an optimal aspect ratio that depends on the details of the confinement?
Okay. Let me try to understand the question better if I understood it properly. Is this the question is if I have an aspect ratio where the cells will perform better in certain confinement, in certain confinement they will not. Is
Right. So
that the question?
if you're competing spheres versus rods. Suppose you have
Right.
shorter rods versus longer rods, you know.
Yeah. So if you come back to this overall phase space, as you can see, like if you increase the aspect ratio beyond certain, like approximately 2, your growth advantage is pretty much similar. And there the advantage, if I go back, it is a little hard to read here because of the scale. But here we are competing E. coli, which is a non-motile E. coli, versus a motile E. hormeisi, Enterobacter hormeisi. So you will also see that E. coli is doing slightly better, and it is probably a competition between a lot of different things. It's not just shape at that point, right? Now, to answer your question, a slightly elongated cell should enjoy a slightly beneficial in terms of confinement, but that is not always true because sanguines is really, really filamentous, but they have other problems because they are very filamentous. They will have to generate a lot of pressure. So a lot of complex things will come. But what is genuinely true is that if your aspect ratio is beyond certain cutoff number, you will actually perform better than the aspect ratio lower than that. This
Mm.
is what we have seen and observed many, many times in many systems.
Okay. Thank you.
So there's another chat from another question from Dip. Great work. Do you have any insights into why all the low aspect ratio bacteria are non-motile and vice versa?
Uh, uh, so uh, that is what we isolated and that is what we had our hands on. Uh, I would really like to do it with a motile spherical bacteria, but unfortunately, what we could isolate from the gut of the red flower beetle were all non-motile. And we
Let's see.
actually, there is also a small thing that E. coli, you can make it non-spherical. But when you make the E. coli spherical, our hypothesis, it becomes non-motile, okay? With A22, it becomes non-motile. It is viable perfectly. It grows perfectly under a shaken culture, but it becomes non-motile. So all of the E. coli studies where we made it sphere, we used a non-motile E. coli to make it to sphere to change the motility completely from the equation. But if you take a motile E. coli and you make it a sphere using A22, it becomes non-motile, probably because it cannot make a bundle in a polar and achieve a polarity, right? Because the flagella are all over the body and they cannot probably bundle more effectively. So possibly that is, I'm not 100% sure about that, but they are non-motile usually. So we couldn't actually test if the motility makes in the spherical system makes a huge deal. But we know for sure that in rod-shaped cells, motility with or without does not make that much difference.